Hey there everyone, so now that we've had a chance to finish our entire unit on the nervous system, which as you're going to recall was all about the communication of stimuli, afferent signals going from sensors to control centers being mostly the brain and the spinal cord, and then efferent signals going from the brain and the spinal cord, carrying that integrated information, carrying instructions about how to maintain homeostasis to a variety of different effectors. So those effectors, more often than not, are going to be some type of muscle tissue. So we talked about how somatic motor neurons are going to carry information specifically to skeletal muscle, and then in a lot of cases, both parasympathetic and sympathetic signals are going to carry information to either cardiac muscle or smooth muscle. So naturally, the next place that we're going to go now that we've finished talking about the entire nervous system is some of those effectors. Specifically, this chapter, we're going to focus on the physiology of muscle tissue. So let's get this out of the way so we can see some of our objectives here. As we've seen before when we first started discussing tissue, there are three different types of muscle. There's skeletal muscle, there's cardiac muscle, and there's smooth muscle. So what we could do with this chapter is we could spend an equal amount of time on skeletal, cardiac, and smooth muscle, but the way that we're actually going to do this is we're going to spend the vast majority of our time talking about skeletal muscle. So what we will eventually find, and we will spend some time comparing and contrasting the three different types of muscle tissue, but what we're going to see is that our discussion that we'll have on skeletal muscle, which will be the most complete discussion that we're going to have, it should cover probably 80 to 90% of the stuff that we need to know also for cardiac and smooth muscle. So really we will see that cardiac and smooth muscle, with some exceptions, are really not all that different from skeletal muscle. So we'll talk most about skeletal muscle. Cardiac muscle we'll talk about a little bit, but since we're just about to start into our cardiovascular unit, we'll get plenty of that later on. And then smooth muscle we'll talk about a lot when we start talking about urinary system, digestive system, and things like that. Okay, so one of the major things that we talked about with the nervous system is that neurons are excitable, and you will recall that when something is excitable, it means that it is capable of changing its membrane potential. So it turns out the same goes for muscle tissue, muscle cells. Whether it's skeletal muscle, smooth muscle, or cardiac muscle, these cells are excitable in exactly the same way that neurons were excitable. They've got a resting membrane potential, and they are capable of opening ion channels in response to a physiological stimulus in order to change that membrane potential so that they can produce an action potential. So almost exactly the same thing that we saw with neurons. So let me get this out of the way so that you can see that. So the stimulation in this case, meaning our EPSPs or IPSPs, are going to come from those motor neurons that we discussed in chapter 15. So muscle cells can either get an EPSP for depolarization or hyperpolarization for an IPSP as the result of neurotransmitter release from either somatic motor neurons or autonomic motor neurons. So that kind of complete lengthy discussion that we had on these different types of neurons, the three different types of neurons, somatic, parasympathetic, and sympathetic, what you're going to see here is the type of neurotransmitter that was released coupled with the receptor that is waiting on the target cell is going to have a great deal to do with whether we get an EPSP or an IPSP, and that will help to determine whether we get muscle contraction or not. So again, let me move this out of the way. So let's start by having just kind of a very brief discussion on the three types of muscle tissue. So this is really the exact same discussion that we had all the way back in chapter 4. So skeletal muscle, you will recall, organizes itself into these long, kind of thin bundles. Each one of these fibers, as they're called, is actually an individual cell. You will notice that each individual cell actually has multiple nuclei. So again, there's no hard and fast rule that every cell in your body has to have exactly one nucleus. Skeletal muscle, it just so happens, has multiple. And then the most unique characteristic of skeletal muscle, as you see under the microscope, is that you see kind of these vertical, kind of alternating light and dark bands, what we call striations. Cardiac muscle is organized kind of the same way skeletal muscle is. So you can notice that cardiac muscle is also striated. You can kind of see those alternating dark and light bands there, although they're not quite as easy to see under the microscope there. So one of the major differences between skeletal muscle and cardiac muscle is that skeletal muscle is voluntary contractile tissue, owing to the fact that they are stimulated by somatic motor neurons. Cardiac muscle and smooth muscle, as we will see in just a second, are involuntary. We can owe that to the fact that they are stimulated by autonomic motor neurons, in addition to something else that we will talk about a little bit later. So in this case, as you see here with cardiac muscle, each individual cell, which is called a cardiomyocyte, is actually connected through cell-to-cell junctions to neighboring cardiomyocytes. We've talked about how one of the characteristics or kind of main features of cardiac muscle tissue is the presence of what are called intercalated discs, these kind of heavy staining vertical lines which represent the gap junctions and other cell-to-cell junctions that join one cardiomyocyte to its nearest neighbor. And then smooth muscle is probably, out of the three types of muscle, it's probably the most different, or the one that has least in common with the other two. It is like cardiac muscle, it's also involuntary. It does not have striations, which is going to be due to kind of the lack of protein organization inside the smooth muscle fiber, kind of different organization than what we're going to see with cardiac and skeletal muscle. You can kind of see an overall lack of organization. The way I always kind of like to put it is these smooth muscle tissue kind of looks like someone just threw a whole bunch of cells together. It doesn't really look very organized. So we will spend a little bit of time towards the end of the chapter talking about what smooth muscle looks like on the inside and kind of what its functions tend to be. But like I said earlier, most of our discussion in this chapter is going to focus on skeletal muscle. So before we get into skeletal muscle in particular, for each of those three different types of muscle tissue, we want to be cognizant of the following properties that muscle tissue tends to have. Number one, and arguably the most important of which, is called excitation-contraction coupling. What this basically refers to is the methodology behind how a muscle cell takes stimulation that it gets from somewhere else, meaning coming from a motor neuron. How does it take that stimulation and produce some type of contractile force? We are aware that all three types of muscle tissue are going to produce some type of contractile force to achieve a number of different physiological means. We use skeletal muscle to move the skeleton around. We use cardiac muscle to pump blood throughout the body. We use smooth muscle to regulate the passage of materials through different places, like in the digestive system, the urinary system, the iris of the eye. We've talked about several examples of that already. But for this, we are specifically saying, how do we take electrical stimulation coming from a motor neuron or somewhere else? How do we turn that into an actual mechanical force that we can use to do work? So the organization of contractile proteins and other cellular elements. So basically, after we have, or really before we have our discussion on excitation-contraction coupling, we want to get a good idea of kind of what the machinery looks like inside the muscle fiber, that machinery that's going to actually produce our force that we want. So this will help us answer questions as to what makes one type of muscle fiber striated and others not, and it'll also help us to appreciate the general function of that particular type of muscle fiber. Contractility, referring to the strength of a contraction, so that's obviously important. Elasticity, basically the idea that once a muscle fiber has contracted, meaning it has kind of shrunk in on itself, it's basically kind of like a spring that you're putting tension on. Due to this elastic property, how does the muscle fiber kind of snap back to its original shape and length when it becomes relaxed again? And then finally, extensibility. Basically, this kind of ties into the elasticity part of it. We want to be able to stretch the muscle to a certain extent. Obviously, you can kind of go overboard with these things, but all of these properties are going to be extremely important to our understanding and overall discussion of muscle in general. Okay, so we are aware at this point that skeletal muscle is stimulated by somatic motor neurons. Now, here's the deal with skeletal muscle, and this will be an important thing to understand for later on when we talk about cardiac and smooth muscle. Physiologically speaking, the only way skeletal muscle can become contracted is if it receives a signal from a somatic motor neuron. There is really no other normal physiological way outside of kind of weird muscle cramps or things like that. There is no real physiological way for a skeletal muscle to become contracted unless a clear-cut message via an action potential comes through a somatic motor neuron. So if you look at this picture, this should look pretty familiar from the end of chapter 14. We have a somatic motor neuron. It's got its cell body in the gray matter of the ventral horn of the spinal cord. It projects its axon out through the ventral root of a spinal nerve, and the axon goes all the way to a particular skeletal muscle, and we release acetylcholine, our neurotransmitter of choice for skeletal muscle. And we've talked before about how there's going to be nicotinic receptors there waiting for it. So here's how things are different as opposed to our types of muscle that are under autonomic control, meaning cardiac and smooth muscle. We just said how skeletal muscle will only contract if it receives a message from a somatic motor neuron. So picture this for a second. What if we completely severed that axon that you see right here? If we sever that axon, there is now no way for this skeletal muscle to receive stimulation. So therefore, we would say that it enters a state of paralysis if we cut off its ability to receive efferent message from the nervous system. Now, muscles that are under autonomic control, like cardiac and smooth muscle, are not limited to such restrictions. If you were to sever, say, the vagus nerve, which carries a lot of parasympathetic information, or if you were to sever several thoracic spinal nerves, which carry a lot of sympathetic information, these muscles will continue to contract at a given frequency. This is what makes them different. These muscles have what is called a basal tone, meaning that these muscles will contract rhythmically again and again and again at a given frequency, regardless of any stimulation that is coming from the autonomic nervous system. What that means for us is that these cells are getting depolarized, but they're not necessarily being depolarized by the nervous system. They are actually getting their depolarization from groupings of electrorhythmic cells called either pacemakers if you're talking about cardiac muscle, or pace setters if you're talking about smooth muscle. So what this means is that if you were to sever those nerves that we talked about that go to the heart or go to some other autonomic target, those muscles will continue to contract. The downside is that you take away the ability of the autonomic nervous system to regulate those contractions. We've already seen how the parasympathetic division can reduce your heart rate. So if you have a lot of parasympathetic tone, a lot of vagal tone, your heart rate is going to be very low. Or the sympathetic division can raise your heart rate. So if you think about it kind of like a seesaw with the sympathetic raising activity and the parasympathetic lowering activity, if you take both of those away, you're not really taking away the ability of your heart to beat and contract. You're taking away your ability to regulate those contractions. You're basically taking away your ability to raise your heart rate or to lower your heart rate. So if you were to sever those either cranial or spinal nerves that feed into a particular autonomic target, you're not necessarily paralyzing that target. You're basically just taking away your ability to regulate that target at all. So as we said, All autonomic innervation is there to do is to either increase the frequency of those contractions or decrease the frequency of those contractions. Autonomic input, whether it's sympathetic or parasympathetic, is not there to give you the depolarization you need for contraction as it is with skeletal muscle. It is only there to make the contractions happen more frequently or make them less frequently. Okay, so now that we've had that discussion, and we'll bring that up again when we start talking about cardiac muscle again, let's talk about skeletal muscle in particular. So every muscle fiber in skeletal muscle, and really with other types of muscle as well, every single what we're called muscle fiber is actually an individual muscle cell. One single individual cell with its own plasma membrane, its own nuclei, its own mitochondria, its own endoplasmic reticulum, everything. So the organization of skeletal muscle is a little bit interesting. The way I kind of like to always think about this is if you've ever seen, I guess those Russian, I think they're Russian, those Russian nesting dolls where you have like this really big doll and you can kind of take the top off and there's another doll inside and then you can take the top off of that one. And then basically after you've done all that, you have all these little dolls lined up left or right and they get shorter and shorter the further you go because all the dolls were kind of nested or hiding inside of each other. So the organization of muscle fibers and skeletal muscle is kind of similar. So if you look at one individual muscle fiber right here, inside of that muscle fiber are quite a few cylindrical bundles of protein filaments that we will talk about here shortly. If you look at all the muscle fibers bound together, if we become less complex here, or more complex here, excuse me, multiple muscle fibers organize themselves into bundles called fascicles, and then fascicles then organize themselves all into a big bundle to make up the whole of what you would understand to be macroskeletal muscle. Let me get this out of the way. So individual muscle fibers receive their connections from the axon terminals of somatic motor neurons, which means that if you look at an entire fascicle here with each one of these representing a single muscle fiber, it would not necessarily be accurate to say that all of these muscle fibers are under the control of the same somatic motor neurons. So a whole muscle can either be fully contracted or partially contracted depending on how many of those muscle fibers are receiving input from their given somatic motor neuron. And we'll have more to say on that here in just a little bit. Okay, so even though a muscle fiber looks really different than any other type of cell that we've seen before, be confident that it has all the kind of things that you would expect a typical cell to have. So obviously it has nuclei, we've talked about that before. It's got basically all the organelles that you would expect any type of animal cell to have. So it's got nuclei, it's got mitochondria, it's got a plasma membrane right here, it's got an endoplasmic reticulum, which will be really important later on. So it's got all that good stuff. It's just because of the kind of unique structure of the muscle fiber, these things are organized in kind of an interesting way, as we're going to see here. And it also turns out that we have some unique terminology that we use specifically when we're dealing with muscle fibers. So the plasma membrane of a muscle fiber is actually now called the sarcolemma, sarco referring to muscle. So this sarco prefix is something that we're going to see quite a lot of. So you can keep calling it the plasma membrane. It's just that if you happen to be doing any reading and you see sarcolemma, I don't want you to be confused. Just be aware that this is what that means. If you see the term sarcolemma, it is referring to this plasma membrane of that particular skeletal muscle fiber. And that sarcolemma is what separates the extracellular fluid from the intracellular fluid. So don't forget that. The sarcoplasm is basically just what we call the cytoplasm. The sarcoplasmic reticulum is what we call the endoplasmic reticulum of the muscle fiber. So like I said, you're seeing this sarco prefix show up a lot. And then as cells that obviously require quite a lot of energy, they are going to have quite a lot of mitochondria. And obviously the answer to the question of for what purpose, mitochondria make a lot of ATP through cellular respiration. Muscle fibers, as we are definitely going to see here, are going to blow through an exorbitant amount of ATP, so we need lots of mitochondria to meet those energy demands. So what you're going to notice here is that if you look kind of dead down the center inside of a muscle fiber, you see these protein filaments which are called myofibrils. So these are the actual contractile protein filaments which are going to produce the force that we need for contraction. If you actually look straight down the barrel of this muscle fiber, you're going to notice that things are kind of cramped, right? It seems like most of the volume here is actually made up of these myofibrils, right? So where do you fit everything else? Where do you fit the nuclei? Where do you fit the mitochondria? Where does everything kind of fit in? And that's a pretty valid concern to have. So a lot of those organelles kind of get cramped in there. We kind of jam them in wherever we can. Especially you're going to see that for something like the sarcoplasmic reticulum, it doesn't really look like any other type of endoplasmic reticulum that we've seen so far. It actually kind of forms this mesh-like kind of pipe-like network that runs in parallel to the myofibrils. And there's actually a very good reason for that we will learn about soon enough. So what we want our focus to be for right now, we can kind of tackle some of those organelles like mitochondria and like the SR as they come along. Let's have our focus for now be those myofilaments, the myofibrils that contain those contractile myofilament proteins that are going to be so important for us. So it turns out there are 4 different types of proteins that we need to absolutely, essentially be aware of as we move forward. One of them is called actin. The other is called myosin. Together, actin and myosin form the 2 absolutely most important major contractile proteins that every muscle fiber is going to have. Muscle contraction is possible because actin and myosin have a terrific affinity for each other. So if you've got affinity for something, you want to kind of glom onto it all the time. Actin and myosin love each other. They love to bind to each other, and that affinity they have for each other is going to be, as we will see, what makes muscle contraction possible. The other 2 types of proteins that we will learn about a little bit later are called tropomyosin and troponin. These are actually regulatory proteins. They are essentially there to make sure that when the muscle is supposed to be relaxed, it stays relaxed. And when the muscle is supposed to be contracted, they basically get out of the way and allow the muscle to become contracted. They are there to make sure that muscle fibers do not contract at inappropriate times. And we will learn a bunch about them later on. So if we actually look at these myofibrils here and the contain actin and myosin myofilaments, they are organized into these kind of units that run from place to place. You will see these units, which are called sarcomeres, lined head to tail down the entire length of the muscle fiber. So keep in mind, each sarcomere contains both actin and myosin. So I also mentioned that the sarcoplasmic reticulum, which you can see here in yellow, runs in parallel down the length of the muscle fiber in parallel to those myofilaments, and we will see later how this is going to be necessary for muscle contraction to occur. And we will see that it's going to have something to do with the fact that the endoplasmic reticulum has a knack for storing up lots of calcium. Remember that? So we're going to kind of see a payoff from that in a major way later on. So it turns out we can actually kill 2 birds with 1 stone here. We can learn about the filament organization in skeletal muscle all at the same time understanding why it is that skeletal muscle appears striated. It turns out that the answer to both questions is the same. So what you're looking at here is 1 sarcomere in a skeletal muscle fiber. So let's go back real quick. That sarcomere is this to here, right here. So that's 1 sarcomere, so we're just looking at a slightly different representation of it here. So a sarcomere is made up of two types of filaments, actin and myosin. So the myosin, we'll start there. The myosin is a very thick protein filament. So it's represented here in this purple color. So because it is very thick and very dense, we sometimes call these thick filaments. And then on the other hand, actin is not nearly as thick or nearly as dense, so we call them thin filaments, and you can see them represented there in green. So if you'll notice here, in this sarcomere, we alternate between regions in which there is only actin, meaning only very thin protein filaments, then we go to very thick, then we go to very thin, then we go to very thick. So the fact that we alternate between thickness and not thickness, thickness and not thickness, tells us that this should show up if we look under the microscope, and that's exactly what happens. If you look at skeletal muscle under the microscope, the fact that you see those alternating light and dark bands, which give us the striations that we're so interested in, that is entirely due to the way that these protein filaments are organized within sarcomeres. That would be one of your light bands, dark band, light band. So any striations that you see in striated muscle tissue tells you that your actin and myosin are organized exactly like this. And you can see here in this more realistic microscope image, you can actually see these striations very clearly, in addition to seeing the multiple nuclei that we've talked about before. But you can see the striations here, and you can say that, okay, if I've got a dark band here, that must be where all the myosin is. And then if you see a light band, that must be where a bunch of actin is, then myosin, then actin, and so on and so forth. So if we're going to really understand how muscle contraction works, we can understand how the organization looks in a relaxed muscle tissue, but we want to understand how we go from relaxed to contracted and back to relaxed again. And it's going to have to do with how the actin and the myosin, how the thick and the thin filaments kind of act together in concert to get the contraction to happen. So first thing we'll do is we'll look at a whole sarcomere, which runs from Z disc, which you can see right here. A Z disc is basically just a vertical alignment of proteins that are meant to anchor the thin filaments in place. So you can see a Z disc there, and you can see the thin filaments protruding out from there, and you have one on each side of the sarcomere. So one sarcomere runs from Z disc to Z disc. From here to here, that is one sarcomere. So you will notice here how we have some regions where we only have thin filaments, some regions where we only have thick, and then some regions where we have a little bit of a mix of both. So we actually have names that we use to kind of indicate which one of those that we're talking about. Okay, we've already talked about that. Okay, so first thing, if you see an area that lacks thick filaments altogether, meaning you're looking at a region that only has thin filaments, we call that an I-band. So an I-band is this region here that only contains thin. So that's an I-band. Everything here is not an I-band, and then there's your next I-band right there. If you have an area that does have thick filaments, we call that an A-band. Now, here is where there is sometimes a little bit of confusion. We just got done talking about the I-band where you only have thin filaments. The A-band does not necessarily only have thick. The A-band, the only way that you should characterize this, the A-band contains all of the thick filaments. We don't care whether there's any thin there or not. Every single bit from head to tail of the thick filament is contained within the A-band. So if you look down here, the A-band stretches from one side of the thick filaments to the other. You'll notice the A-band contains a region that is only thick filament, but also regions that contain both thick and thin. The A-band is allowed to contain some thin filaments. The only necessary thing that we use to define the A-band is that it completely contains the thick filaments. So just want to make sure that we understand that. Now, if you're interested in the region that only contains the thick filament, that is the H-band that you see right here. So the H-band, by definition, will only contain thick filaments, so no I-bands are there. Yes, so the H-band accounts for the region that has only thick filaments and no overlap from the thin. And then, as I already said, in the center of each I-band is that Z-disc. It's basically just this vertical alignment that anchors the thin filaments in place. Hang on just a second. My laptop is running out of juice, so let me just plug it in here so we don't lose this video halfway through. There we go. Okay, sorry about that. Okay, so we've already talked about I-bands, A-bands, H-bands, and Z-discs. So that is going to be fundamental to our understanding of what actually happens to the sarcomere when we go from a relaxed muscle to a contracted muscle. Okay, so I think I mentioned this before, but the way I always like to think about about muscle in general is it's kind of like a spring, right? So if you've got a sarcomere, you've got Z-disc here with all your filaments in between. What's going to happen when a muscle contracts is that those Z-discs are going to get pushed together. So any muscle fibers that we have, any contractile proteins that we have, actin and myosin, are going to slide together like this. And what we're going to do is increase the degree to which we have overlap between thick and thin. So that H-band is going to get shorter and those I-bands are going to get shorter. So really it's an awful lot like taking a spring and pushing on either side. You're putting tension on a spring with the understanding that the more you get that spring to contract, the more force it's going to come back with when it kind of springs back into its initial length. So if you look at this picture right here, you can see kind of a electron microscope image here that actually shows you physiologically what's happening in a muscle fiber with two sarcomeres that are visible here. So Z-disc to Z-disc, Z-disc to Z-disc, or if you'd rather look at the more cartoony version over here, you'll get the same information from either one. Whichever one you want to focus on is fine with me. So on top here, whether you're looking here or here, that is going to be what the sarcomeres look like in a relaxed muscle. So you'll notice that the Z-discs are at their maximum length apart from each other, and then you have your I-bands in which you only have thin filaments, your H-bands where you only have thick filaments. As the muscle becomes progressively more and more contracted, we are going to force these Z-discs together. So in particular, watch what happens to your I-bands and your H-bands. So as we push those Z-discs together, look at the H-band. The H-band is getting smaller and smaller because we are increasing that overlap that we get between thick and thin. There is now a smaller and smaller space that those thick filaments have only to themselves. And the same is true for the I-bands. Because you're increasing overlap, those thin filaments have a smaller and smaller space to call just their own. And then once the muscle reaches full contraction, you'll notice that the I-bands have all but disappeared and the H-band has pretty much completely disappeared. We have maximized the amount of overlap we get between thick and thin filaments. We have shortened the sarcomere, and that is going to give us a lot of mechanical energy that we can use to do a lot of different things in the body. So I've already mentioned this for the most part, but you do want to pay attention to what is happening to each of our zones here as the muscle gets shorter and shorter. The only one of the things that does not actually get shorter is the A-band. So the A-band, again, by definition, is just the area that contains thick filaments. We're not actually shortening the thick filaments themselves, we're just kind of forcing them to overlap more and more with the thin filaments. So that doesn't really change the A-band at all. But you did notice that the H-bands and the I-bands, the areas that only contain one filament or another, they do get shorter. So they are going to appear to kind of disappear as the muscle becomes progressively more and more contracted. Okay, so I took note of the fact that one of the most important and fundamental things to muscle contraction, and we will see this is true for any type of muscle fiber, whether it's skeletal, cardiac, or smooth, is this interaction between actin and myosin. I said actin and myosin love each other. They've got this natural affinity for each other. So let's discuss that a little bit more in depth. So actin and myosin, they've got this great natural working relationship because they bind each other with great affinity. So here you can see myosin. Myosin is particularly interesting because it has kind of two major parts to it. It has these heads right here that contain both an actin binding site, basically the part of it that likes to grab onto actin, and then a ATP binding site that allows it to bind to ATP and extract the energy out of it. Similarly, actin has a myosin binding site. So think of it kind of like shaking hands with someone. So you've got a hand that you would use to shake hands with someone else. Obviously, you're not doing that right now because of the coronavirus, so definitely don't go shake hands with someone to prove my point here. But you have a hand, it's your adapter that you use to match the hand of someone else. So you have a hand, they have a hand, myosin, actin, they can grab each other like this because they each have binding sites for each other. And then I mentioned that myosin head also has an ATP hydrolysis domain. Without getting into unnecessary detail here, basically what that means is that the myosin can bind to a high-energy ATP molecule. It can hydrolyze it to extract all that energy out of it. And you can kind of see where we're going with this. We're going to use that energy to power muscle contraction. It's going to be that energy that ultimately forces the Z disc together and gets the muscle contracted. So, a lot to talk about with that. So, the actual process of shortening the sarcomeres is called a power stroke. The way it works is that ATP hydrolysis is actually, let me kind of make this a little bit bigger so you can see this visual demonstration I'm going to put on. The myosin head kind of cocks itself back. It's kind of like a hammer on a firearm, like a pistol, right? So it kind of leans back like this with that ATP energy. It leans back, it kind of cocks itself back into a high potential energy configuration. It basically leans itself back, and then it's going to spring forward once it grabs onto that actin, and that will provide the energy of forcing the thick and the thin filaments together. They're going to slide in opposite directions, so we force them together. So the thing to take note of here is that it's the ATP hydrolysis that cocks that myosin head back into this configuration that allows it to move forward with a lot of energy in order to get the power stroke to happen. So the myosin head, when it binds to and uses ATP, it cocks itself back, and then all that has to happen after that is it has to grab onto actin, and everything else with the power stroke should happen kind of naturally. And then just to kind of give you some perspective here, every muscle fiber is going to have millions, billions, if not trillions of myosin heads that are all each going to require ATP molecules to get all of this done. So that's a lot of ATP, a lot of energy that we're going to need. So hopefully you can appreciate why aerobic respiration in the mitochondria is so important. You don't really want to get yourself in a situation where your muscle fibers run out of energy. That's how muscle fatigue happens, right? And we'll have a good lengthy discussion on that here in a little while. And then it's also worth noting that while we're on the topic of energy, muscle fibers, muscle tissue in general, tends to receive a very rich blood supply. So what's the connection between ATP and the blood supply? It's oxygen, right? So aerobic respiration in the mitochondria is only possible with oxygen. With oxygen, we make about 36 to 38 molecules of ATP per glucose molecule utilized. That's only 2 if you don't have oxygen. So very important to get oxygen delivered to high energy tissues like muscle fibers. So here's a question for you to kind of think about, and you can, if you can't quite settle on an answer to this, maybe bring it to our Collaborate discussion that we either have on Wednesday or Thursday, depending on which class you're in. If you're talking about muscle fibers needing a lot of energy and their utilization of ATP has gone up, meaning you're really contracting those muscles and their demand for energy has gone through the roof, how could your body possibly go about increasing the amount of blood delivered to skeletal muscles? So you're going to want to think about something that we talked about in the previous unit. So think about it, come to our Collaborate session on Wednesday or Thursday, and see if you've got the answer to that. And I'll give you a hint here because I forgot that this slide was coming next. You're going to want to use this slide right here. So think about this slide right here. Think about the fight or flight response that we talked about before. Try to come up with a way that explains how something like this would either increase or decrease oxygen delivery to muscle fibers in general. And then you'll want to obviously know, is this a sympathetic response, is it a parasympathetic response, or could it possibly be both? So make sure that you kind of understand that as you move forward. All right, so actin and myosin, two of the four types of major proteins that you find within sarcomeres. We said that actin and myosin are absolutely the most important, and we've had a good discussion on that. But what about troponin and tropomyosin? All we really said about them before is that they are essentially there to make sure that the muscle fiber will only contract at the appropriate times. So let's kind of get this straight in our heads given what we've talked about. So far we've said that the myosin head here on this entire thick filament, the myosin head will bind to ATP, hydrolyze the ATP to extract the energy out of it, and then it will cock itself back into a high potential energy configuration. From there, the only thing that has to happen in order for contraction to occur is that cocked back actin, or excuse me, that cocked back myosin has to grab onto actin, and then it slides it forward automatically from there. So this begs a question for us. What is it that keeps muscle relaxed? So the interesting thing that I didn't really tell you before is that even in relaxed muscles, so go ahead and relax your body as much as you possibly can. In all of your relaxed muscles right now, your myosin heads are already cocked back and ready to go. They have already utilized their ATP to cock back into place. So the question is, what is it right now that is keeping your muscles relaxed? Knowing that those myosin heads are cocked back and ready to go, all they need is they need to grab onto the actin. So what is keeping your muscle relaxed right now? Well, it turns out what's keeping your muscle relaxed is these regulatory proteins that we're just now talking about, troponin and tropomyosin. So let's learn a little bit more about them. So tropomyosin, what you see here in this kind of pink color right here, tropomyosin is a long filamentous protein that runs in parallel to each of the actin strands. And specifically what it does is it lays on top of those myosin binding sites. Essentially what it is doing is it is blocking the myosin head, which is cocked back and ready to go. It is blocking it from having access to where it's trying to grab. Imagine if you reach your hand out to shake someone's hand, and trying to come up with a good analogy here, say they reach out to grab your hand, but they're holding like a big metal ball or something. I can't think of a better example than that. They reach out to grab your hand, but they're already holding on to something. You can't shake their hand if they're already holding on to something. So that's essentially what tropomyosin is doing. It lays on top of the place that myosin is trying to reach out and grab. So even though the myosin head is cocked back and ready to go, it has nothing to grab onto as long as tropomyosin is blocking those myosin binding sites. It blocks the formation of cross bridges. So this is what keeps your relaxed muscle relaxed. The fact that tropomyosin is being pesky and getting in the way of myosin grabbing onto the actin. Like I said, in all of your relaxed muscle right now, those myosin heads are cocked back and ready to go. It's the tropomyosin that keeps your muscle relaxed. Well, with that in mind, the question then becomes, okay, how do we go from relaxed muscle to contracted muscle? Obviously, we are going to have to get that tropomyosin out of the way. So things are going to stay like this until this other protein that you see right here called troponin, which is actually a complex of several proteins. Things are going to stay relaxed like this until troponin can get its hands on calcium. Troponin is a calcium-binding protein complex. What it's going to do will all depend on whether it has calcium or not. Here, when troponin has no calcium, it's not doing anything. It does nothing. Tropomyosin is still in the way, so nothing changes. Once troponin binds to calcium, troponin is going to pretty much wake up, and it is going to grab tropomyosin and shift it, move it out of the way, so myosin is now free and in the clear to grab onto actin. Tropomyosin that is bound to calcium takes that tropomyosin and shifts it away from those myosin binding sites, so now that myosin, which is cocked back and ready to go, it has something to grab onto now. But here is the tricky thing. This is what we're going to have to try to get straightened out. You will recall the intracellular fluid has hardly any calcium at all. Calcium is maintained at such a minuscule concentration inside the cytoplasm, or in this case sarcoplasm, that really it would not be that inaccurate to say there's no calcium at all. So as long as things are just kind of the way they normally are, troponin's not going to be able to come across any calcium. So the question we want to answer here, where can this calcium potentially come from? Where can we possibly get calcium to just suddenly show up in the intracellular fluid? Because what we can understand here is that the only way we are going to go from a relaxed muscle to a contracted muscle is if we can somehow find a way to increase the intracellular calcium concentration. So where can we possibly get that calcium to come from? So your first inclination, and it's not a wrong inclination certainly, but your first inclination is probably, okay, so if there's not very much calcium inside the cell, which you can see is definitely the case here at 1/10,000th of a millimolar, which is hardly anything, what if we just took some calcium that is in the extracellular fluid? You can see it is much, much, much more highly concentrated out there. What if we open a calcium channel in the sarcolemma, in the plasma membrane, right? So if we do that, calcium can move down its concentration gradient from high concentration out here to very, very low concentration in here. So that would definitely do it, right? So if we open a calcium channel, whether it's a ligand-gated one, a voltage-gated one, or whatever, if we open a calcium channel, that would increase the availability of calcium and troponin could bind to it. That's certainly not a bad idea, but that's not quite the way it's going to go. We are going to get our calcium from somewhere. It's just not going to be the extracellular fluid. Do not forget, the endoplasmic reticulum, we talked about this way back in Chapter 3, and I promised you that we would see the payoff for it when we started talking about muscle. The endoplasmic reticulum, in addition to being involved in a lot of protein synthesis with all its ribosomes and lipid synthesis, if you're talking about the smooth endoplasmic reticulum, the endoplasmic reticulum stores up and hoards a lot of calcium. So a big reason why there is hardly any calcium in the cytoplasm is because through active transport, the endoplasmic reticulum spends ATP to gobble up all of the calcium for itself. So we basically store up a good majority of the cell's internal calcium stores in the endoplasmic reticulum. And we store it up all for situations like this. If, with a physiological stimulus, such as this G protein coupled receptor that you see right here, if we can somehow use a second messenger system to trigger the opening of a calcium channel in the endoplasmic reticulum membrane, we can have calcium flooding out into the cytoplasm so that it can bind to troponin and it can stimulate muscle contraction. So let's have just a little bit of a review here very quickly for what we'll call excitation-contraction coupling. And I've already posted a separate review video, which I think is maybe like 10 or 15 minutes long, so you can look at that if you want to kind of review this whole thing here. So, number one, calcium ions have to be made available in the sarcoplasm, and we do this through the opening of calcium channels in the sarcoplasmic reticulum membrane. So that's what we've covered so far. So we need to finish the rest of this excitation-contraction coupling. Okay, we understand that calcium binds to troponin, that gets the tropomyosin out of the way, that allows myosin to bind to actin, and that gives us our power stroke. But what about all the stuff before that? In talking about that, we seem to have skipped a whole bunch of steps, right? So, like I said, calcium binds to troponin, that gets tropomyosin out of the way, and then blah, 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 all the stuff that we've talked about already. But what was the stimulus that caused the calcium to be released, right? So that's something that we haven't really talked about yet. So let's go ahead and do that. So we're aware, and we're kind of bringing ourselves full circle here, we're aware that the somatic nervous system, through those somatic motor neurons, they exert a great deal of control over the contraction of skeletal muscle. Remember how I said skeletal muscle will not contract most of the time unless that signal comes from a somatic motor neuron. We're talking about anything but basically muscle cramps. So how do we link these things together? How do we link the activity of those somatic motor neurons to the contraction of muscle and all this calcium stuff and troponin and tropomyosin stuff that we've talked about here? So let's go back to the very beginning. So what you're looking at here is a single muscle fiber, so that should look pretty familiar. And here, what should also look familiar is the axon terminals of a somatic motor neuron. So if we take a little bit of a closer look at one of these axon terminals here, or what are sometimes called synaptic boutons, you can see the axon terminal of this somatic motor neuron, and then you can see the sarcolemma of the muscle fiber. This is the synapse between the somatic motor neuron and the skeletal muscle fiber. We have a special name for this. It is called the neuromuscular junction. Specifically, that synaptic bouton of the somatic motor neuron is accommodated in kind of a invagination in the sarcolemma, which is called the motor end plate. It's basically this kind of hollow area that is meant to accommodate that axon terminal. Specifically, the sarcolemma at that motor end plate contains a lot of nicotinic cholinergic receptors. Do you remember what kind of neurotransmitter that receptor binds to? Well, you win the prize if you said acetylcholine, right? So acetylcholine is released by the somatic motor neuron here into the neuromuscular junction, and then there are nicotinic receptors there waiting for it. So one of the first things that has to happen if a muscle fiber is going to contract is that acetylcholine has to bind to nicotinic receptors on the motor end plate of the muscle fiber. So what will be the result? What did we say always happens with nicotinic receptors? We said nicotinic receptors are always excitatory, so the result is always going to be depolarization. So the interesting thing, we know from our discussion on neurons, we know that depolarization, EPSPs, that's a good thing if we want an action potential. It's the same thing here. Depolarization gets us closer to a threshold that we need to get to fire an action potential. But here's the deal. Axons of neurons are what were carrying action potentials then. Skeletal muscle fibers do not have axons, so we need to kind of answer the question of what is going to carry the action potential here, because we definitely don't have axons. So if the muscle fiber does get depolarized enough to fire an action potential, it will propagate through the sarcolemma. Specifically, it's going to go down these little rabbit hole pathways called T-tubules or transverse tubules. The transverse tubules actually lead directly into the sarcoplasmic reticulum. Specifically, because that pathway from the sarcolemma directly to the SR exists, that allows us to get the depolarization to the sarcoplasmic reticulum membrane, and that is going to allow us to open voltage-gated calcium channels on the SR membrane. So you can see here exactly how we go from depolarization of the muscle fiber from acetylcholine to the release of calcium from the SR. So once these voltage-gated calcium channels open, they release their calcium into the sarcoplasm, and that binds to troponin, and everything else should be hunky-dory from there. So you'll also notice here that we've said before, the sarcoplasmic reticulum is located in very close proximity to those myofibrils. So we want to make sure any calcium that gets released is immediately going to get gobbled up by troponin. So here is a useful slide. Again, I've said this a bunch before. I would definitely bookmark this slide if you are into studying this process step by step, which I highly, highly, highly recommend. So this covers step by step, in chronological order, everything that happens with excitation-contraction coupling. I'll just briefly go through it one more time, and like I said, I've got a review video that's already posted for you to look at. So somatic motor neuron will release acetylcholine as its neurotransmitter. That will bind to nicotinic receptors on the motor end plate. That will always produce depolarization, as we've discussed before. That should get us an action potential, which goes through the transverse tubules to the sarcoplasmic reticulum. We open calcium channels. Calcium floods into the sarcoplasm. Calcium binds to troponin. It moves tropomyosin out of the way. Myosin, which is all cocked back and ready to go, binds to the actin, and we get our power stroke. We shorten those sarcomeres, and the muscle produces a contraction that gives us the contractile force we need to do whatever it is we are going to do. So I think we're going to make this a two-part video. So I'm going to go ahead and cut things off here. So check back again. I will have part 2 of chapter 10, I think it is we're on right now. I'll have part 2 of this coming up. So we will cover everything from muscle relaxation to muscle fatigue, different types of muscle fibers, and then we'll finish off our discussion with cardiac and smooth muscle. So we'll go ahead and sign off here for now. I'll see you back for part 2 here in a little bit.